Okay, good afternoon. I'm Fred Gilman, Dean of the Mellon College of Science and Buhl Professor of Theoretical Physics, and it's my pleasure to welcome you to the 2014 Buhl Lecture. This is the 19th Buhl Lecture in this series, which was reinstituted in 1996, and it's the third time that the topic has been biological physics. In fact, one might describe the topic of these three lectures as biochemical physics or biophysical chemistry, or whatever other way you want to phrase the three fields together of biology, chemistry, and physics, because this is the area where those three subjects overlap. And we're very pleased to have been able to bring today one of the leaders in this multidisciplinary area, Carlos Bustamante, to Carnegie Mellon and give this year's Buhl Lecture. Bustamante's laboratory has developed and continues to develop ways to manipulate single molecules using optical and magnetic tweezers. Using such tools as group twists and stretches DNA molecules, analyzes their elasticity, unfolds individual protein and RNA molecules, and understands how complex molecular machines actually work. What a fantastic and wonderful workshop to be playing in, and to in fact have helped to open this whole field of research. Professor Bustamante grew up in Peru, received his undergraduate education there, and then he came to UC Berkeley for his PhD in biophysics. As befits somebody with this multidisciplinary approach, he's now a professor of molecular and cell biology, professor of chemistry, and the Raymond and Beverly Sackler Professor of Physics at UC Berkeley. So he has feet, or chairs, more particularly in each of three departments. He's been a Howard Hughes Medical Institute investigator since 2000. He's a member of the National Academy of Sciences, won the Hans Neurath Prize of the Protein Society, the Wilczek Prize, and the Raymond and Beverly Sackler International Prize in Biophysics, among many other honors. So I invite Carlos Bustamante to give today's Buhl Lecture as he tells us about the work being done in that has been done and is being done in his laboratory, and also discusses how his studies have benefited from parallel developments in statistical physics to learn about the energetics of complex molecular processes. The title of his talk, as you can see, is appropriately Biochemistry and Biophysics, One Molecule at a Time, When Less is More. Carlos?
Thank you. Thank you very much. Thank you very much, Fred. Thank you all for coming. I've been fighting a cold, and they told me if I took these pills, I would not get a cold. And so far it's almost true, except that I'm losing my voice. So forgive me if I cough during my talk. I have some water to help me here. And so today what I would like to do is to tell you work that have been done in my laboratory for the last about 20 years or so, and in particular give you a sense of the process that how one finding led to another finding, and so on and so forth. So the first question whenever you actually talk about doing single molecule biophysics is why single molecules? And here is one of the reasons. The truth is that the type of molecules that we study in biophysics are molecules that can exist in more than one form, oftentimes at equilibrium in different forms. And so if you're lucky, in fact, it's possible that if you measure the average property of a molecule, this property corresponds to a distribution which is present in solution that is narrow. And so the average, in fact, reflects well the situation you have in solution. But it's possible that actually you have rather a broad distribution in this molecular property, and the average doesn't really reflect what's going on in solution. And in some cases even, you may actually have, for example, a bimodal distribution in which the average doesn't reflect at all what really is happening in solution, the average of this particular molecular property. And so the first point that I want to make today is that averaging is not a good idea. Oftentimes can be very misleading. And I found a little poem written by a Chilean poet, actually, some years ago. I have it here in English and Spanish. Nicanor Parra, who says, there are two breads, you eat both, I eat none, average consumption, 1 bread per person. So, averaging can be very misleading, and the point today that I'm going to try to make today is that averaging also in biophysics and in molecular biology and in biochemistry and even in cell biology is usually a bad idea. And I want to emphasize more the point in the following way. It turns out that one place where this microscopic, let's call it microscopic, single molecule, few molecules, view matters is the cell interior. Why? Because we know that many cellular processes, such as chromosome replication and segregation, DNA transcription, recombination, RNA translation, are often carried out by very few molecules, sometimes even one single molecule. And ultimately, we want to know how things happen in the cell. And so, for example, studying replication with 10 to the 18 molecules in a tube doesn't quite give you the sense of what happens when a single DNA polymerase starts copying the leading strand of the bacterium, for example, and copies the whole strand. And that's all the DNA polymerases that are involved in copying the leading strand. So if you really want to know how does the cell perceive the process of replication, you are better off if you are in a position to really follow this type of processes one molecule at a time. Let's take a bacterium, in fact, like E. coli, and let's take a single molecule of any protein, of any molecule if you want. We know the volume of E. coli is about 1 micron cube, okay, about a micron by a micron by a micron. If you calculate what is the concentration of that molecule in the cell, you'll find that it's 1.6 nanomolar. Just one molecule is at the concentration of 1.6 nanomolar. And you know that many proteins, for example, that bind to DNA, they tend to bind with subnanomolar affinity, meaning that a single molecule, which is at 1.6 nanomolar, is sufficient to ensure that 99% of the time that binding site in the DNA will be occupied by that molecule. In other words, the cell doesn't need to synthesize 3,000 molecules of that particular protein in order to ensure binding to the active site, to their binding site. Okay, so many years ago now, I was a professor at the University of New Mexico in Albuquerque, in the chemistry department, and we became interested in a very simple question. We know that electrophoresis is a very powerful method that biologists use all the time to separate molecules. And we wonder how is that molecules actually get separated inside the gel. And I start asking around, and actually nobody knew. The fact is that everybody was using the method, but nobody knew how it happened. So I thought it was an interesting question to answer, and we designed a very simple experiment. In this experiment, what we did is we grabbed a piece of, a microscope slide, and we mixed some agarose gel with molecules of DNA that had been mixed with a fluorescent dye, a dye that when it intercalates in the bases becomes fluorescent. So we mixed the fluorescent DNA with agarose on a slide, microscope slide. We put a cover slip, we seal it with nail polish, that's always good to have around, in the laboratory, and then we put 2 electrodes, and then the gel hardens, and we observe the following. This is real-time molecules of T for DNA moving towards the positive electrode, as you see here. Here is the positive electrode. Both arms extend towards the positive electrode, but just by chance, one of the arms is longer than the other, has more charge, experiences more force in the field, and pulls the other end around. See? This is how DNA molecules move inside the gel. But what really surprised me was the fact that when it pulls, it's pulled around the corner. Look how it catches up. It's very elastic. The molecule of DNA is very elastic, and I have taught elasticity in my class, but I have never seen it. And here it is, right? So I was really shocked by the elastic nature of the molecule, and I said, wouldn't it be fun if we actually could grab one of these molecules by the ends and pull the molecule and decide and measure the elasticity of the molecule. Here is one last. There you go. So, and that was the idea. The idea was grab a single molecule of DNA, and I guess we didn't know enough to dissuade us from trying to do the experiment, and eventually we did the experiment. And so the idea was attach a molecule of DNA to a wall, and then pull the other end with a force, and then determine the extension and elasticity of the molecule. In practice, the way we did it is we attached a molecule of DNA to two beads, two small beads, one atop a micropipette, the other we actually grab it with an optical tweezers machine, and then we apply force and we extended the molecule. The actual first experiment was done with magnetic tweezers, not with optical tweezers. So I want to be clear about that. But the idea was to determine the force needed to extend the molecule a given amount. This is a physics 1 experiment where you are just measuring the spring constant of spring by essentially applying higher and higher forces to the spring. And here is the data. We publish this force in the y-axis extension, and you see 4 molecules being extended. They all look very similar, luckily, and you see that DNA is a highly nonlinear spring. Namely, the molecule doesn't follow a linear relationship between force and extension. This is Hooke's law, right? And in fact, this is the way Hooke expressed his law. He said, "Uctensio sic vis," which means, like the extension, so goes the force. Okay, so one is proportional to the other. And so we were able to actually characterize this. And at the time we did this experiment, it was not immediately clear to us that we had the basis to do a number of other experiments. We were just pulling molecules of DNA, and it was fun, and we published it, and okay, fine. But all of you who are here are scientists, and you know that that's the whole point, right? To have fun. But as it turns out, actually, there were other things that could be done, and that became possible thanks to this characterization, because eventually we were able to treat mathematically, in collaboration with John Marco and Eric Silla, the problem of the extension elasticity of DNA, and we finally ended up obtaining this simple expression that relates the force that you must apply to the molecule to the extension that you obtain when the contour length of the molecule is L and its persistence length is P at temperature T. Okay? So now we have expression that allow us to convert at a given force the end-to-end distance or extension of the molecule to contour length. And that turned out to be the basis of a number of assays that are being used today to follow many processes, biochemically interesting processes. But as I point out, at the time where we did this work, we were still blind about the possibilities that this will open. Now, I want to say that this model is a wormlike chain model, and it has become very useful because As it turns out, it works not only with double-stranded DNA, but also with single-stranded DNA, with polypeptides, with sugars, with polysaccharides, and so it's very, very useful in general. And here you can see a comparison between the two leading theories for the elasticity of DNA, the freely jointed chain in dashed line, and the wormlike chain that I just showed you in a continuous line, and the data. And you can see that the data follows very nicely the wormlike chain model, and not so the freely jointed chain model that describes the DNA as a polymer made out of rotational independent Kuhn segments. So we became bolder about it, and we decided to actually try our luck in other areas. And we said, what would happen if we would actually grab a molecule of RNA and pull it using this ability to manipulate molecules? And we were very ambitious. We actually used the group 1 intron from Tetrahymen Thermophila. You know, this is a ribozyme, this is an enzyme RNA, right? It adopts tertiary structure, and so we decided we were going to study first this simple part that is shown here in red. We call it the P5ABC hairpin. It has a 3-helix junction. And this molecule, as simple as it is, this part of the molecule, is quite complex. So we decided to get rid of all the red bases that you see here, the bulge and the helix in red, and even do a simpler molecule, which we call P5AB. And see just what happens if you pull this molecule from the ends. We extended this molecule a little bit more with more RNA, and then we put a piece of DNA, handles on both sides, so we could actually grab the molecule. And under the optical tweezers, now we are doing this with optical tweezers, we were able to follow force versus extinction. Because the handles of DNA have their own elasticity, at the beginning you will see that there is a monotonic behavior due to the handles. But at some point, the molecule is going to open at some force, and that's what we were after. So you will see here that, in fact, the molecule opens at about 13 piconewtons, and when we relax the molecule actually refolds at about 13 piconewtons also. So in fact, the unfolding that we are obtaining is a reversible unfolding. It's force versus extension, so the area under the curve is work. So we can measure the reversible work for unfolding the molecule, which happens to be identical to the change in free energy of the molecule, as thermodynamics tells us. So in fact, from doing this experiment, we can calculate thermodynamic free energies for the folding or unfolding of the molecule. But we also noticed the following. When we were pulling the molecule, sometimes we would be pulling, the molecule will unfold, and before we could do anything else, the molecule would refold and then unfold again before we continue pulling. So we thought this was interesting. So we said, what would happen if we get the molecule very close to the unfolding force and hold the tension constant at that force, and tell the machine, the optical tweezer machine, keep the tension constant no matter what. Okay, so what will happen is that the molecule is at the verge of making the transition, then a fluctuation will occur which will lead to the unfolding of the molecule. The tension will tend to zag because the molecule becomes longer, but the machine won't allow the tension to change, so it moves the bits apart. Then the molecule decides to refold. The tension will increase. The machine won't allow that to happen, so it will bring the bits closer. And then what you observe is that the machine is doing this, right? So you can actually follow the opening and closing of the molecule. And in fact, here you see the distance between the bits versus time at 12.6 piconewtons. You see that the molecule is most of the time in the closed form and transiently visits the unfolded state, right? Now, we are going to tilt the equilibrium by applying a little bit more tension from 12.6 piconewtons to 13 piconewtons. And now we make the molecule spend almost roughly 50% of the time in the folded state and 50% of the time in the unfolded state. And finally, with 0.2 piconewtons more, now you see that the molecule is most of the time unfolded and only transiently visits the folded state. Now, because we can repeat this and follow this for a long time, we can determine the average time the molecule spends in one state and the average time it spends in the other state. And from that, we can obtain the rate constants as the inverse of the time, the rate constant for unfolding and for refolding. Some of you may have seen actually this data, much nicer and beautiful now from Aumita Mandal's laboratory, but this is the first experiment that we did. So you notice the background, now in Aumita's laboratory, this is perfectly flat. This is perfectly flat, it's fine, beautiful, okay? But it was the first one, so actually our background noise, our drift was not very well controlled, but I thought I'd show you the original data for honesty. So, the important thing about this is that from here we can get rate constants for unfolding and refolding. The ratio of the rate constants gives me the equilibrium constant for the process at a particular force, and I can always extrapolate to zero force and obtain the free energy that everybody could relate at zero force. Right? So we have here that, in fact, we show that an expression similar to Van Hoff's equation exists, except that now it's not the temperature dependence of the of the equilibrium constant, but it's the force dependent of the equilibrium constant, which gives you the size of the molecule once it unfolds. And in fact, we determined that the free energy at zero force for this molecule is 156 kilojoules per mole, which is very close to the value predicted from the program called mFold. Okay? So we can get thermodynamics and we can get kinetics out of this type of unfolding or refolding processes. And the length of the molecule is 23 nanometers. Okay. It turns out that you notice that we also can get the rate constant as a function of force. Well, an equivalent to Arrhenius equation occurs, but again, is the derivative with respect to the force of the rate constants or the rate parameters. And now a new parameter appears. Notice the distance to the transition state. Why? Because for the first time you are imposing one particular coordinate along which the molecule is being unfolded. And along that very well-defined coordinate in space, you can ask, where is the transitional state? So you can measure the distance to the transitional state. And we found that for this molecule, the distance to the transitional state was halfway between the closed form and the open form. Okay? The molecule is very compliant. Good. And here is the data, the logarithm of the equilibrium constant, of the rate constant as a function of force, and the other constant, the rate for closing the molecule, going from open to close, of course, decreases with force. So, I just showed you this result. What happens if we add these things? Well, it turns out this molecule, in the presence of magnesium, can adopt tertiary structure. Not only secondary structure, but tertiary structure. Well, immediately makes things a lot more complicated. Notice that when we unfold the molecule, we see an intermediate. The step of unfolding occurs in two steps. In this case, first step, second step. And when we relax the molecule, actually, the process does not occur at equilibrium. We get hysteresis. The pulling curve is in blue. Here is unfolding. The refolding curve is in green, and the refolding occurs at a force much lower. We are having hysteresis in the presence of magnesium. And we know that this characteristic is because the molecule is adopting tertiary structure, because if we take away the magnesium by adding EDTA, a chelating agent that will grab the magnesium, the molecule now becomes reversibly unfoldable. We lose a lot of the characteristics and mostly the hysteresis. Okay. So it turns out that it's possible still to get information about the thermodynamics of a process carried out of equilibrium by using new theorems that have been discovered in the last 15 years called fluctuation theorems in statistical mechanics. But I'm not going to tell you that today because tomorrow I have to give a talk, if I have a voice left, that precisely talks about the use of these fluctuation theorems to extract free energies from non-equilibrium processes. It sounds like an oxymoron, free energy from non-equilibrium, which is to say equilibrium property from non-equilibrium experiment, but that's precisely the novelty of what I'm going to tell you, that these new fluctuation theorems allow us to actually get equilibrium information from non-equilibrium experiments. Okay, nonetheless, we were able to, using this expression, obtain the distance to the transition state, and now we get 1.6 nanometers. Remember that the other was 10 nanometers, halfway? Now the molecule is harder, as soon as the molecule adopts tertiary structure, the molecule is harder to break, and it actually is, as soon as you deform it a little bit, it breaks completely. So it becomes brittle. And this is a characteristic of all tertiary structures, both in proteins and in RNAs. When the molecules adopt tertiary structures, they become brittle. Okay? Meaning, is the difference between bending a garden hose, which bends smoothly, and bending a chalk. As soon as you actually start deforming the chalk will break, right? And the process is irreversible, in fact. So the analogy is correct in that sense. So this is one of the things that we have found by actually playing with these molecules. But I want to show you something that is more interesting about this. We became bolder and we said, let's pull the whole molecule, okay? And when you pull the whole molecule, you get all kinds of transitions. And my student actually carried out this work. And I want to show you, unfolding the full ribozyme, what happens when you pull a molecule as complex as this. It turns out that the molecule will undergo transitions, but will not always unfold by the same pathway. Sometimes we will unfold one thing first, other things next, and we can actually do it over and over and over and determine the potential energy surface over which the molecule diffuses when it goes from the folded state to the unfolded state. And that's what you're going to see here. This is one example. First transition, another. So it went this way to the unfolded state. Now we repeat the experiment. And you see that the molecule is going through all this intermediate before going to the transitional state. In other words, we can actually see the different pathways the molecule adopts in going from the folded state to the unfolded state. Here is an anecdote that happened when we did this work. The postdoctoral that it was a fellow that was doing, student that was doing this work, her name is Viviana Onoa. She's from Colombia, but when we talked, we never talk in Spanish, we talk in English. Obviously we could speak in Spanish in principle, but actually we talk in English. And she came and showed me some of this work, and I said, you got it. Now what you have to do is pull this molecule 800 times, and you'll get all the data that you need, because you will be able to tell the probability of going this way or that way, whatever. So she left, and 2 weeks later she came back and told me, okay, you said 800 times, I pull it 900 times. And I said, 800 times? I said 800 times. And so you see, actually her Spanish ears and my Spanish accent led to this mistake. But a good postdoc always makes the right mistake, right? She pulled it 900 times, and the result of that is this map that gives us the thickness of the arrow, tells us the probability that the molecule will go this way or that way or that way, quantitatively. In other words, we have essentially the equivalent to a mapping of the potential energy surface over which the diffusion of the unfolding of this molecule occurs. All right. So now I want to switch gears and tell you about the work of Marcello Malpighi, who in 1666 wrote a book called De Viscerum Structura. And he says in this book, the operative industry of nature is so prolific that machines will be eventually found not only unknown to us, but also unimaginable by our mind. Marcello Malpighi was the leader of the revolution that was occurring in the 17th century in Europe in the biological sciences. He was a professor at the University of Bologna and a professor of microscopic anatomy. The previous century had been the time of macroscopic anatomy, but now they all had microscopes. And so he was a professor of microscopic anatomy. And in fact, he was leading this revolution that was occurring in the biological sciences, which paralleled the revolution that was occurring in physics. In fact, here is the book, where Galileo and Newton were working and in fact defining the concepts of force and acceleration and inertia, terms in which eventually we will cast the description of how machines work. In both cases, these men were helped by the invention of instruments that allowed them to see things that the naked eye could not see. In the case of the physicists, the telescope. In the case of the biologists, the microscope. These instruments and this technology revolution essentially was the fuel that forwarded the revolution in both disciplines. So Malpighi was thinking in 1666 about machines, tiny machines that are unimaginable by our mind, he said. So, in fact, the idea of the mechanical paradigm is not new in biology, and it recurs. It has been recurring over and over. As late as 1998, where Bruce Alberts, then president of the National Academy, wrote a very influential article in Cell, where he said the entire cell can be viewed as a factory that contains an elaborate network of interlocking assembly lines, each of which is composed of a set of large protein machines. Okay? So, in fact, the idea is that the cell has a modular factory. We no longer think of the cell as a little bag of a concentrated solution of nucleic acids and proteins that is undergoing second-order reactions. We now think more of it as this factory made up of these machine units that are responsible for all these functions that you see here, right? Chromosomal segregation, transcription, splicing, translation, protein folding. And these machines, in fact, are ultimately molecular machines. So, in fact, it is possible then to exploit these methods that I've been telling you about, because these methods actually allow you to measure force, to apply force, to measure torque, to apply torque, and to measure distances, which are precisely the variables that you want to describe the behavior of machines, right? Many of them are in fact molecular motors, capable of converting chemical energy in the form of ATP or a gradient of an ionic solution into mechanical work and force and torque generation. So here in this case, I'm going to show you one example. This is Pol2. This is the enzyme responsible for reading the DNA and converting it in a molecule of RNA. And we can actually grab one of these molecules, just as shown here. We grab it by the back into a tiny little bead, which is trapped in an optical trap, and then the distal end of the DNA over which the molecule wants to walk, the polymerase wants to walk, is also grabbed in another bead on another trap. And now when we add the MTPs, the nucleotides that the molecule needs to synthesize the RNA molecule, shown here in red, the molecule cannot move because it's attached to the bead. So what it does is pulls the DNA through itself. All motion is relative, but in so doing, it's pulling this bead out of the trap, and that allows us to follow transcription in real time. This process called transcription in real time. And so here you see the length, the end-to-end distance, And notice that the position I label here is in nucleotides, namely the number of bases that have been walked by the polymerase. But I only measure end-to-end distance. But remember, we know very well that formula that allows us to go from end-to-end distance to contour length. So in fact, I can express the end-to-end distance at a given force into contour length, which is in this case expressing nucleotides. And you see that after playing tug of war with a single molecule, we can see how the molecule behaves. It's transcribing, then for no apparent reason it stops. Then it starts again, then it stops. Then it starts again, then it stops. Starts, stops. So we've discovered that there is a lot of interesting dynamics in the behavior of this protein, and I don't have time to, in this general lecture, to give you an explanation of what's going on here, but lots of processes, important biological processes, are related to this process that the molecule makes. In particular, the molecule actually goes backwards during this process. Notice here? The molecule is going backwards, meaning that the molecule is moving rearwards, and the reason is the molecule is trying to edit its own copying. And if it makes a mistake, it's able to correct the mistake by cleaving the RNA if it made a mistake and incorporated the wrong base. But to do that, it has to pause and move backwards, okay? And we can follow this movement directly in real time. We can ask a question, and what happens when the polymerase encounters a nucleosome? Our DNA is all wrapped in nucleosomes, and the question is, what happens when the polymerase arrives and encounters this enormous roadblock? Well, we asked that question, and here is the experiment I just showed you, and here is the experiment where a nucleosome is put in the path of the polymerase. Notice that I grabbed here the DNA upstream of the protein, so the protein is going that way, and that the tension that I'm applying is helping the polymerase. But notice the difference in behavior between this case and that case. The molecule passes through the nucleosome part, where there is no nucleosome, largely unaffected, 87% of the time. Whereas in the presence of the nucleosome, notice that the molecule, as soon as it encounters the nucleosome, stops for a long time, and then manages eventually to pass. Sometimes doesn't even pass, only 58% of the time. So in fact, you see that in the presence of the nucleosome, the molecule has many long pauses and pronounced backtracking during those pauses. So, in fact, we learned many things about how the polymerase works. Here's another anecdote. My students finished the paper, sent it to Cell, and it was accepted in the journal Cell. And then we decided we were going to send a front cover for the article, and they built up this in plastic. You can see that actually, right? You can see the polymerase is going there, and the nucleosome is telling it stop. And I thought it would never be accepted by Cell, but they did something else. They actually animated it. They animated it and made a video. And in the video, you see the polymerase moving, arriving, the nucleosome putting the stop sign, raising the stop sign, and then the polymerase still goes on, and then the nucleosome opens up, his face, and happy, and then eventually the polymerase goes around it and recuperates it. When I saw the video, I said, it is 99% sure that you are going to get the front cover. And they did get the front cover for the Cell because of that. So, for example, we learned the following, which is very interesting. The enzyme actually is not strong enough to peel the DNA from the nucleosome. But what the enzyme does is it arrives to the nucleosome and sits there. Then the nucleosome has an opening due to fluctuations. And as soon as it opens, the polymerase moves in. And then sits there again, waiting for another fluctuation. And so in fact, the RNA polymerase rectifies the fluctuation of the nucleosome, and that's how it actually crosses the barrier. And then we show that there is a number of elements, these tails and other things, that actually control the magnitude of this barrier that the nucleosome imposes on the polymerase. Okay, so let me show you one more example. Here is the process of translation. Once the mRNA has been synthesized by the polymerase, now a new machine, the ribosome, copies the RNA into protein. And that's the one that ultimately expresses the genetic characteristic, right? So if we say brown hair or blue eyes, you need the gene for the brown hair, for the color of the eyes, and that gene is carried by a protein of a particular characteristic, which is actually coming out from the ribosome by reading an RNA. We wanted to study the process of translation, one codon at a time. We did this work with Nacho Dinoco and with Harry Noller. So here is the trick. You grab one of those hairpins that I showed you of RNA, okay, and you pick these ends with optical tweezers, with beads, of course, and now the ribosome will bind to the base of the hairpin, and then you add all the elements that you need to do translation: initiation factors, elongation factors, amino acyl tRNAs, everything that you need, the ribosome needs to move and synthesize the protein. As it moves, it will open the hairpin, right? You will have to open the hairpin. As you open the hairpin, it will actually the molecule will get longer, right? Will get longer, and if you are measuring the distance between the beads, the distance between the beads is getting longer, right? So you can follow translation at the single molecule level. Here is the experiment. Here is the hairpin, here is the ribosome loaded at the base of the hairpin. Now, as we add the amino acyl tRNAs, this is what you observe in the optical tweezers. Notice that these dashed lines are separated by 3 base pairs. So 3 base pairs are open every time. Sometimes we miss one, and of course when the hairpin gets too small, it's too unstable, and then it opens completely. But notice this part. You see that, in fact, we can see the ribosome stepping one codon at a time, namely 3 nucleotides, 3 nucleotides, 3 nucleotides, or 3 base pairs are open in the case of a hairpin. So we can actually follow the actual process. We call this the helicase assay, because you have to break a helix, open the helix. The ribosome has to open the helix in order to advance. But we can also do a tug of war geometry, because now we grab the ribosome by the back, and then we grab the distal end of the RNA, and now we are playing tug of war with one single RNA, a ribosome, and then the ribosome has to move along the RNA, and we can actually see that very clearly also under the tweezers. Notice these are separated by 3 nucleotides, these dashed lines. Notice how noisy and fluctuating is the signal. This is single molecule. In bulk, all of this is averaged out, and you just get translation, but you don't get the dynamics of the machine actually doing the work. And now I can play tug of war, really, and apply more or less force to the molecule and determine, for example, the rate of translation as a function of the force that I apply. So here I decrease the force, and then I increase the force, and I see how the rate of translation of the ribosome change. In other words, I'm studying the motor properties of the ribosome. And here is what we obtain. Translation rate, how the translation rate decreases as I increase the force. It turns out that the rate of decrease gives me information about the distance to the transition state. which happens to be 1.4 nanometers from this data. And the step size of the motor, we just measured it, is 3 nucleotides or 1.7 nanometers, which shows that in fact, just the fact that more than 50% or 60% of the step is the distance to the transition state means that the ribosome does the whole movement in one single step. No, it's not 1, 2, 3 for each nucleotide, but it's a single 3 nucleotide step what the ribosome does. And then you can do other type of experiments. I show you the ribosome translating, but what is the product of translation? The production of a protein, right? And here is an interesting point, an interesting question. A molecule like T4 lysozyme, this is a protein that has 160 amino acids. The ribosome will take 8 seconds to synthesize the whole protein. But the T4 lysozyme molecule can fold in only 50 microseconds. Okay? So I want to emphasize again. It takes the ribosome 8 seconds to synthesize the molecule, but the molecule can refold in only 50 microseconds. So here is the question. Since the synthesis of proteins occurs vectorially, one amino acid at a time, what makes or prevents the protein that is coming out slowly to adopt a folded structure that may kinetically trap the molecule, right? Instead of waiting until the whole molecule has come out to adopt the native structure dictated by the full sequence of the protein. So that's a question. And we wanted to answer that question. And so we thought maybe we can answer that question by comparing the folding or unfolding of the protein on the surface of the ribosome, coming out of the ribosome, versus a protein that is in solution, just in solution. The same protein in solution, without the ribosome. So here it is, the protein in the absence of the ribosome, just in solution. We're going to pull it and refold it. And this is what we observed. The molecule unfolds, refolds, unfolds, refolds, unfolds, refolds, every time. Very sturdy, very strong, right? Resilient. You pull it, unfold it, relax it, refolds. Now we repeat the same experiment, and now we are pulling the protein from the surface of the ribosome. The first time we pull, nothing happens. We don't see unfolding, as if the protein was not folded to begin with. Then, the next time we pull, immediately we pull, we unfold, we see the unfolding, but we don't see refolding. And then we pull, no unfolding, no refolding, no unfolding, no refolding, and so on and so forth. Finally, the molecule manages to fold, and then we see the molecule refolding. It turns out that the presence of the ribosome surface slows down the refolding of the protein 200 times. And in fact, we know that it's the electrostatic interaction between the surface of the ribosome and the protein. What is unfolded is retaining the protein. The protein is coming out and is being grabbed, so to speak, by the surface of the ribosome, and actually is prevented from folding so that as not to get kinetically trapped in a structure that from which the molecule can then not proceed to the folded state, to the native state. Finally, and come to the end, I talk to you about two examples that we've been studying of a series of motors that are being studied by many people, but also in my laboratory, which are called ring atpases. Ring atpases are a huge superfamily of proteins in the cell that are responsible for carrying transport properties. For example, when E. coli wants to segregate, copies the DNA before division in order to have two cells, two daughter cells. Each daughter cell needs a molecule of DNA, otherwise they won't survive. There is a machine that is a ring ATPase called FTSK that pulls one molecule to the right side and pulls the other molecule to the left side, okay? And that is a ring ATPase that actually does that by burning ATP, okay? And when the cell wants to actually transport molecules across a membrane, it uses one of these ring ATPases to do the transport. So I'm going to tell you about two of them, because this superfamily is divided in two clades. One is called the Rec A clade, and the other is called the AAA plus clade. And I'm going to tell you about one example of the AAA plus clade and one example of the Rec A clade. The first one, the GlypXP protease, it belongs to the AAA plus clade. And this work has been done with Andreas Martin, a professor at UC Berkeley in my department, MCD. Okay, so here is the question. What happens when the cell wants to get rid of a protein that is no longer useful to the protein, to the cell? Now you say, why would the protein not be useful to the cell? Well, there are many reasons. Some proteins are synthesized just to activate processes, and once you get that process activated, you don't want it anymore, when you don't want that process to continue occurring. So the cell has to get rid of the protein. Or the protein gets oxidized, and it doesn't function well when it is oxidized. So the cell wants to get rid of the protein. How does it get rid of the protein? This is a moment of poetic beauty for me, because when I used to go around the country saying we are studying how proteins fold or refold by pulling them, there was always a question asking, excuse me, what is this to do with the way a protein folds or unfolds? Meaning that we were not using urea or temperature to unfold our proteins. But the only thing I could answer was, well, to my knowledge, the cell, when it wants to unfold the protein, doesn't squirt a little bit of urea to the protein or raises locally the temperature to unfold it, right? That's what biochemists do. And they do that because they cannot use force to unfold. Well, now we know that the way a cell unfolds a protein and gets rid of protein is by using a molecular machine, which is a protease, which is an unfoldase. In this case, it has many. In fact, E. coli has 7 of those machines. And what it does is this. When you want to get rid of this particular protein, you put a little tag at the end terminus called an SSRA tag. And this SSRA tag is recognized by this ring ATPase that grabs the tag, burns ATP, and pulls and unfolds the protein mechanically by force, and then passes it into a second chamber where there are proteases that cut the amino acid polypeptide and releases the new amino acids to be recycled, okay? So the unfolding of proteins is in fact mechanical in the cell and carried out by molecular machines. And so here is the experiment we did. We have a GlypXP attached to a bead in an optical trap. Then we have another bead here that carries a handle of DNA, 3 kilobase pairs, and that at the end has a green fluorescent protein, an unfolded titin, a green fluorescent protein, and a little SSRA tag in front. And so what we do is, with an optical tweezers, we go like that to the other bead. It's like enticing. You all have done this, right? I mean, try to get the spider to come out of the nest by actually tweaking it with a little, well, that's exactly what this experiment is. We go like that, and we try to actually make the GlypXP to come out and start fighting, right? So, anyway, so that's what happens. And so what you see here is what we observe in real time. the ClipXP grabs the SSRA and immediately moves in front of the green fluorescent protein and start pulling, right? And then you see what happens here. We are measuring the distance between the beads, so the size of the complex. There, it unfolded the first green fluorescent protein and keeps and processes it, and here it unfolds the next green fluorescent protein and moves it. You see? So the protein is pulling here and eventually manages to unfold the green fluorescent protein. Actually, we now know that there is an intermediate that actually is always populated here, right? And you see it here again, okay? So, and then once it unfolds the first green fluorescent protein, it just processes it and chops it. And then until it gets to the front of the second one. And when it gets pulls during all this time, until eventually it manages to unfold it and to actually process it. Okay, so you can measure the velocity versus force, because we can, of course, apply more or less tension to the molecule, and you see that this molecule, this motor, can generate forces as high as 20 piconewtons, which is sufficient to unfold most proteins in the cell. Okay? So, and here is something very interesting. When you look at high resolution, how the actual process of pulling is, you find that the motor works in two phases. There is a phase where the motor doesn't do anything, which we call a dwell time, and then there is movement, translocation of the polypeptide. Okay? And then again, dwell, translocation. We call it dwell and burst, dwell and burst, dwell and burst. And here is what we found, very interesting. The burst size, how many amino acids or how many nanometers does the molecule move after the dwell, varies. Sometimes it's 1 nanometer, 2 nanometers, sometimes 3 nanometers, sometimes 4 nanometers. Here you see in blue. Okay? Now you lower the concentration of ATP, the fuel, and then you see that the 4 nanometers disappear, and mostly you get 2 nanometers. In other words, if you lower the concentration of ATP, the burst size gets smaller. What about the dwell time? The dwell time is invariant. So it's very interesting. This motor works as a clock. Once it binds the first ATP, it's a matter of constant time before it now eventually starts translocating. So it has a constant dwell but a variable burst. And the constant dwell is like an internal clock that is telling the motor, go and unfold. Now, why does it go from 4 to 2? Because if there is little amount of ATP in the solution, only 2 of the subunits, of the 6 subunits, can bind ATP. So only 2 of them move. Whereas if there is a lot of ATP, 4 of them can actually bind ATP and move 4 nanometers. Each subunit moves 1 nanometer. In other words, we have constant dwell time, variable burst at different ATP concentration. The reason I mention this is because I'm going to show you an example where exactly the opposite happens in a different motor. And this motor is one that sits at the base of a virus called Phi29. This virus infects bacteria, and then the capsid self-assembles inside the bacterium. But now the problem is, how do we get the DNA inside? Well, there is a tiny little machine at the base of this motor that actually grabs the DNA of the virus and burns ATP and pulls the DNA and packages it inside the capsid. Now, you can do this experiment. Now you are familiar with the type of experiments we do. So here is the capsid attached to a bead. The end of the DNA is grabbed to a bead and in another optical trap, and now you can measure the distance between the piece of DNA outside, the DNA tether, that has not been packaged, as a function of time. Notice it comes down. If you increase the concentration of ATP, the motor does it faster and faster and faster and faster, right? The slope is the rate of packaging, about 100 base pairs per second of DNA, so it's very fast. And when you look at high resolution, again you see dwells and burst, dwells and burst, dwells and burst. So now we are going to vary the concentration of ATP. We are going to go from high concentration to low concentration, or vice versa. Notice, the dashed lines here are separated by 10 base pairs. And there is a dwell, then 10 base pairs, dwell, 10 base pairs, dwell, and so on. If I increase the concentration of ATP, notice the dwell times now get shorter, shorter and shorter and shorter, as I increase the concentration of ATP. But the dwells, but the bursts, are always 10 base pairs. So it's exactly the opposite to the other machine that I just telling you. Now, if you increase the concentration of ATP, the dwells get shorter, but the bursts don't change, don't get larger. If you decrease the concentration of ATP, again, the bursts are constant, 10 base pairs, but the dwells get longer. Okay. What is the step size of the motor? Well, every once in a while we could see things in between, indicating that the motor is really sub-stepping. There are 5 ATPases in this ring. 5 identical ATPases. We measure, we apply a lot of tension and measure it. We have an optical tweezers machine that has a resolution of 1 Armstrong at room temperature. So we can actually follow the actual stepping of the motor. And it is 2.5 base pairs. Now the dashed lines here are 2.5. Notice the pairwise distribution function between any 2 pairs of points in these curves gives you a maximum of 2.5, 5, 7.5, 10. Okay? So, in fact, when my students came back and told me, here is the answer, it's 2.5 base pairs, I said, no way. It's supposed to be 2 base pairs, according to people that have done the studies in bulk. And you are telling me 2.5? You know, biologists don't like non-integer numbers. It has never been possible to actually publish that the step size per ATPase is 2.5 base pairs. Go back, I don't believe it. So they went back and repeated experiments and then came back and said, in fact, you were right. They said, it's not 2.5, but actually 2.4 plus minus 0.1. Which incidentally is the standard error of the mean in this experiment, which means that it's 0.3 of an Armstrong, is the standard error of the mean. And since there are 10 base pairs in a burst, it follows that 4 2.5 or so base pairs make a 10 base pairs. But I told you that there are 5 ATPases in this ring. So what happened with the 5th ATPase? That's the question, right? Like 5 identical ATPases. And now I tell you that it moves by 10 base pairs made up of 4 power strokes of 2.5 base pairs. So what happened with the 5th ATPase? So, to make a long story short, we suspected that one of the 5 ATPases is special and adopts the identity of being a special subunit, and its function is to regulate the other 4 ATPases. Okay? How we know that? We know that because some years ago we grabbed a molecule of DNA that was being packaged by the motor, and we replaced a piece of DNA by chargeless DNA, methylphosphonate DNA, which doesn't have a charge. And the motor had trouble crossing this chargeless DNA, this neutralized DNA, particularly if the phosphate that had no charge was at a distance of 10 base pairs from another phosphate that had no charge. So the motor was having trouble making this the motor needed to make a phosphate contact. And then we said, maybe the subunit that makes the phosphate contact every 10 base pairs becomes special by making the contact with that phosphate. How could we show that this is the case? The answer came out from experiments that had nothing to do with this, as often happens in science. We were asking a completely different question, which was, does the DNA rotate as it packages inside the capsule? In other words, does the motor induces force and torque as it actually packages the DNA? And to answer that question, we were doing this experiment. Here is the phage, here is the DNA, here is the bead, the pipette, and we put a tiny little bit on the side of the DNA. If the DNA gets rotated as it gets packaged, we will see this bead rotate around, right? And so here is the movie of that experiment. You see that as the packaging proceeds, the little bead is rotating, indicating that the DNA, in fact, this machine, this molecular machine, is pulling the DNA and rotating the DNA, apparently. Okay? So, this was very nice, but we found the following, that the amount of rotation per base pairs package varied with the amount of DNA inside the capsid. And immediately, someone came and told me, I bet the motor is not rotating the DNA. I bet what you are seeing is that because the DNA is organizing as a supercoil, as a solenoid inside the capsid, the DNA outside the capsid has to rotate in order not to plecton in, not to form a supercoil, which has more energy. The system will try to minimize its energy, and that, therefore, it actually rotates. So it's a passive rotation. I can demonstrate that here, maybe this way, right? So here is the DNA, right? And now it's going to be packaged in a solenoid. And see what happens to the bead, the rotor bead, when I package in the solenoid. See? It rotates. So maybe the rotation we are observing is related to the organization of the DNA inside the capsid, not due to the actual torque exerted on the DNA by the motor. How can we prove one from the other? Well, prevent the DNA from organizing like this inside the capsid. What can we do to do that? So, well, in Peru, my country, in the museums, you see lots of these type of heads because the Incas had developed, it says here, Peru has produced some of the largest samples of prehistoric trepanated individuals. Since trepanation was practiced over a long period of time and across a broad geographic area, the Cusco trepanation survival rate reached 90% at one point and was accompanied by a low frequency of infection, 4.5%. This is from the Journal of American Physical Anthropology, 2008. We know that the people survived because they put a plate of gold to relieve the expansion of the brain when a soldier got hit in battle. The brain swelled and that killed the person, so they opened a hole and then they put a piece of gold and the bone grew over the gold. The gold was gone because the Spaniards got there first, but the bone had grown over the gold, and so that means that the patient survived. So the idea was obvious. Why not actually produce trepanated heads of bacteriophage? And we found that by freezing and thawing the heads of the bacteriophage, they develop heads, holes in the head, basically. And here is the normal bacteriophage head that has not been trepanated. As it packages, it slows down. This is distance versus time. It slows down because the internal pressure of DNA inside the capsid slows down the motor, right? So the slope slows down, is getting smaller. Here is a trepanated prohead. Constant velocity, because obviously the DNA is going one way and coming out the other way. So now we ask, does the DNA get rotated with trepanated heads? That's the question. And the answer is yes, it does. It does, and it is minus 1.5 degrees per base pair. Minus 1.5 degrees per base pair. So you say, what is minus 1.5 degrees per base pair? I told you the motor packages in increments of 10 base pairs. Dwell time, 10 base pairs first. Dwell time, 10 base pairs first. Right? Okay. Let's assume that the special subunit just made a contact with the phosphate, then tells the other subunits package. They package 2.5, and now it wants to contact this phosphate. But is the phosphate there? Well, it turns out that the periodicity of DNA is not 10 base pairs, it's 10.4 base pairs. Right? Which means the following, that in order to encounter the phosphate exactly where the special subunit needs it, every time, it will have to package the DNA by 10.4 base pairs, but the motor packages it by 10 base pairs, which means that the phosphate is not where it should be, because it has not gone around 360 degrees, but it's less than a full turn for the DNA, so it's only 346 degrees, which is 14 degrees shorter than where it should be. So the motor, every 10 base pairs, rotates the DNA by 14 degrees for the special subunit to contact the phosphate. 14 degrees in 10 base pairs is 1.4 degrees per base pair. And we measure 1.5 degrees per base pair. So we think that in fact, what we are observing here is the result of the special subunit being special and needing to contact the phosphate in order to coordinate and tell the other subunits package the DNA. We know this is partly true because we found that as the pressure increases, of DNA increases inside the capsid, the motor changes its step size. And from 10 degrees, 10 base pairs at 0% of packaging of the DNA gets down to 9 base pairs at 99% of packaging. Obviously, if you are changing the burst size, you will have to change proportionally the angle. Look, this is the change in burst size, and this is the change in angle. So in fact, this motor wants to preserve its coordination and will change the rotation angle as it changes its step size in order to maintain the mechanism of coordination. So, to complete the story, I'm going to show you the final model of this motor. At the beginning of the cycle, it's full of ADP. The special subunit loses the ADP. Binding of ATP kicks the ADP out. Binding of ATP kicks the other subunit ATP, so we combine ATP, binding of ATP, leaving ADP, binding ATP, and now the special subunit, which has a different color, hydrolyzes but doesn't translocate until the other subunit start hydrolyzing and translocating. 2.5, 2.5, 2.5, and now the special subunit says, where is my phosphate? Rotates the DNA and releases the phosphate, ATP binding, releases ADP, binding of ATP. We know this is interlaced, we've shown that. Binding of ATP, releases ADP, and now the special subunit does the first hydrolysis without translocation and tells the other subunits, hydrolyze and translocate. Hydrolyze and translocate, and then now rotate the DNA 14 degrees. And again, starts all over again. All right? So that's the story. And here are the people that have worked with me throughout all this work. I may have missed one or two, it's nothing personal, but I should point out Ignacio Tinoco, my ex-research advisor and current collaborator, still working, we're working together, Doug Smith, Andreas Martin, and Susan Marquesi, who are also colleagues of mine at Berkeley. Thank you very much.
think in the hour, we should go directly to the reception, which is 2 floors up in the atrium of the Mellon Institute, and we get a chance, I'm sure, to ask some questions of Carlos there. Let's thank you again.